Hena, application scientist from Refeyn, and today I'll be speaking about de-risking gene therapy development and stress testing viral vectors and with mass photometry analysis. We'll go through the stress testing and its importance, how you can utilize the mass photometry and macromass photometry for it, through some experiments that we have done in-house on adeno-associated viruses and lentiviral vectors, and how we use our two instruments, SAMUX and Caretro, to analyze those samples. And at the end, how mass photometry and macromass photometry can help you de-risk your gene therapy. Stress testing plays a crucial role in understanding how a product behaves under some challenging conditions. And it's not always about finding when a product degrades, but why and what is driving those changes that can affect its quality, safety, or efficacy. So it's important to assess the product's stability, but also its degradation pathways and what type of degradation products are generated during this process. Are they potentially harmful byproducts? For example, when we expose viral vectors to some elevated temperature, we may see that capsid starts breaking down, and this immediately tells us that temperature control is critical. But it's not only about stress testing and understanding the degradation pathway, it's also by understanding what are the optimal conditions for those viral vectors and how we can optimize the process and the formulation by identifying those critical quality parameters and selecting excipients and defining the storage conditions. Also, these studies can help us understand product weak points early on, allowing us to design and support a better analytical method development and ensure the product's quality through its shelf life. And in the end, the stress testing will give us the data that we need to ensure the product integrity through its shelf life to provide strong science-backed evidence for regulatory submissions. Now, once we understand why stress testing is important, now the question is, how do we do it? And what are the different types of stress testing? So each give us some unique window into product behavior. So for example, let's start with a temperature stress that, for instance, will help us evaluate thermal stability. We can see how quickly product degrades at some elevated temperatures or some very, very low temperatures or after a few freeze thaws. And this can help predict a long-term stability of the product. On the other hand, pH stress testing that allow us to identify the ideal pH range or where that pH induces degradation, which will give us some sort of window for the ideal pH for the formulation. And then there is a light exposure, which is essentially for detecting a photosensitivity. Some viral vectors might be affected by the light, and that is also important for the storage conditions or maybe even excipients that are being used. Oxidative stress tests will help us understand how vulnerable these products are to oxidation, which is particularly important for the formulation. And then we have finally a chemical degradation to identify the breakdown products and some degradation pathways. Once we move into, well, the viral vectors, stress testing then becomes much more challenging because unlike other biomolecules, viral vectors are very complex biological systems that, and we know that each viral vector behaves differently. LVVs are inherently different to AAVs, and then even within AAVs, each serotypes behaves differently. They do have different melting points, just as one of the characteristics. So the methods need to be very, very carefully optimized. And this makes these type of tests very time intensive with multiple complex steps and a very, very long turnaround times. That means that we will end up with limited throughput and scalability. And because these assays require significant amounts of materials, sample and resource demand can also become a bottleneck. So how can mass photometry and macromass photometry address these problems that were listed previously, and can this method address all the needs for analysis of stress-tested viral vectors? I will just give you now a brief overview of what mass photometry and macromass photometry is before going into the experiments and explaining why this method was relevant for testing these viral vectors. Again, so mass photometry measures the mass of single particles by light. So when the analyte is immobilized in solution, is immobilized on the glass slide, and the laser shines light through, these particles scatter the light. But there is also a light reflected from the glass surface, and that interference of the light produces something known as contrast. So that contrast is directly proportional to mass, and to be able to convert it, we use our mass reference B2 calibrant of known mass to convert contrast into the mass. So in case of AAVs, the output looks like the picture on the right-hand side, where we can measure mass of the empty partial for any normal filled capsids and distinguish them clearly. Then when we move to macromass photometry, besides the mass photometry side of it and measuring contrast, which is a proxy for a mass, in this case we are introducing also this Z-sweep or vertical sweep, which then allows us to measure the size and diameter of the molecule. So the output is actually two parameters: the size of the particle and the contrast, which is the proxy for mass. And how that, so how measurement on the macromass photometer or Caretro looks in real life. So this is just an example of a size reference or our calibrant for Caretro MP, and that is an equimolar mixture of silica beads that have 5 different diameters. So in the case of Caretro MP, we can clearly resolve those 5 distinct population based on the 2 parameters, which is size and the contrast. But in case where someone uses other methods such as NTA or DLS, that will cause a bias toward a larger size. Now I will walk you through experiments and stress testing of adeno-associated viruses and lentiviral vectors. For adeno-associated viruses thermal stress experiment, we used externally purchased USB standards and varied temperatures, and we incrementally increased them from 65 to 75 degrees, and varied incubation times for 5, 15, and 30 minutes. And then we analyzed those samples using 2 types of glass slides: mouse glass UC, which are the type of glass slides that I usually use for measurement of AAVs, and mouse glass NA that I usually use for measurement of nucleic acid. And the reason why we use mouse glass NA will become clearer as I explain what we did in our experiments. So just to give you the idea, these were our control samples that we used to determine empty and full ranges. So for full, we measured it at the room temperature. So what happens when we expose these samples to a thermal stress? As you can see, the samples measured at a room temperature have 90% of the full AAV capsids. But when they're exposed to 65 degrees for just 15 minutes, that percentage falls to 49% of full, and there is a clear shift in these population. By increasing that incubation time further to 30 minutes, there is an additional increase of 9%. Or if we then further increase temperature to 68 degrees for the same amount of the time for 15 minutes, we see a further decrease in the full AAV population to 17.3. So we see the significant shift of those population, which is our first indication that So what happens if we keep increasing the temperature and get close to, slowly start getting closer to the melting point of AAV8? So for the shorter period of time at 70 degrees, we can see that the genetic payload leakage and that population, again, population shift in full and empty, and around 28.9% of full. But there is some difference in the pattern where we can see a distinctive partial population of AAVs. To ensure that our capsids are not degrading and what we see is an actual payload leakage, we had to test negative controls to indicate that the capsid remained intact at these tested temperatures. So therefore we tested empty AAVs at the same temperatures, and they remained stable throughout, with like a small peak of 620 kilodaltons started forming at 72 degrees, which is already indicating that maybe capsid started degrading and that we are getting very close to 72, 73. Which is considered to be the melting point. Now, the reason why we use mouse glycinate for nucleic acids is because with these type of slides, we are able to distinguish a population that corresponds to a single-stranded DNA. So beside a decrease in the full percentage that we saw already by testing these samples on mouse glass UC and seeing that shift in population, this is the confirmation of the payload leakage, because the peak around 890 kilodalton that is formed, it does correspond to the expected ssDNA value, which was predicted to be around 873. And with a longer exposure or increase of the temperature, the intensity of this peak becomes higher. But then what happens if we keep increasing the incubation time at these high temperatures? Now we do see, again, that peak that corresponds to single-stranded DNA, but we can also see the degraded AAV capsid and ssDNA peak overlap at one point where we exposed our AAVs to extreme temperatures for 15 minutes. And in this case, also there are some hypotheses that the leakage in nucleic acid is causing a formation of aggregates, and we can see also some aggregates forming as well. So a brief exposure to 70 to 72 degrees ejects that ssDNA from the capsid, and that peak intensity increases with the temperature and with the exposure. But with prolonged incubation, we can see the capsids start progressively degrading, and that peak of single-stranded DNA and degraded particles starts slightly overlapping. Now, the genome presence, again, is validated by the lack of a peak in control samples, which were our empty AAVs that we tested that they remained stable and consistent throughout. And then a small peak appeared after 30 minutes at 65 degrees. So we know that a melting point of the AAV8 capsid is around 73 degrees, but we wanted to test and bring those AAV capsids to complete degradation, and they do undergo complete degradation at 75 degrees. So we can't see anything in the region where we are expecting our AAV capsids, but we certainly see two very distinct peaks. And then the benefit of using Mars glass NA is that we are able to see our nucleic acids or our ssDNA peak in the region that we expected, but also we are able to see our degraded AAV capsids within the region that the mass region that we expected. Now, it is important to caveat that due to the use of MFP2 for calibration, that ssDNA appears slightly higher in molecular mass than the calculated and expected value, but this is going to be further optimized. So it is very nice to see the two peaks that really corresponded to degraded capsid proteins, and then they don't overlap with how nucleic acid and the full degradation took place. Now on to lentiviral vectors. So we used slightly more gentle conditions for thermal stress experiment of lentiviral vectors. These were also externally purchased lentiviral vectors, and we incrementally increased the temperature from 37 to 50 to 60 and 70 degrees. But we also tested how freeze-thaw could potentially affect lentiviral vectors. The incubation time was varied from 10, 20, and to 30 minutes. And for the analysis, we used our instrument or macromass photometer, Caritra MP, and the mass glass KV, which are consumables for the measurement. So it's important to understand that lentiviral vector preparations are very inherently heterogeneous, and as such, they do require gating. So that gating is determined or based on the negative control. So we are able to show here the gates that correspond to our target molecule, which are our lentiviral vectors, or the gate A, and then we have gate B, which are our host cell proteins, exosome, cell debris, or any impurities that were generated in the process. And because this is also a size and contrast measurement, we know that the lentiviral vector size is like from 80 to 120. And the smaller the size, that means that some of these lentiviral vectors are either degraded or they're immature. So this is the gate for them. When we tested these samples, this is your Caritra MP analyzed sample and the scatter plot generated. And by calculating the physical titer using the instrument software, we determined that the physical titer is 2.5 e to the 10 particles per milliliter. We already had a value for the TU titer, which was 5.18 e to the 8 TU per mil. And this was determined using functional titer facts. And the ratio between them was 48.26. Now, what happens if we expose the sample that you already saw to 37 degrees? So in this case, the blue scatter plot represents our lentiviral vectors pre-treatment, and then going from orange, gray to green, that incubation time at 37 degrees increased. In case we are able to quantify the change in each of the population of your in gate A lentiviral vectors, in the gate B your host cell protein impurities and low molecular weight impurities, and whether we see any increase in degraded viruses. And we do. When you're looking into gate A and the corresponding line on this chart, we see that there is a decrease in lentiviral vector population from 35 to 61% from the starting value. But there is also an increase in molecular weight or in low molecular weight impurities from 9 to 13%, and there is a clear degradation happening that is almost 120% higher. So prolonged incubation and at this temperature would potentially even further damage lentiviral vectors that are very sensitive to as we know, the temperature and shear and and a lot of other environmental conditions. But what happens when we expose those viral vectors to even higher temperature of 50 degrees? So again, your blue scatter plot is your sample pre-treatment, and then you can see samples that were exposed for 10, 20, and 30 minutes. And again, gate A corresponds to your lentiviral vectors, and gate B and C to lower molecular weights and degraded capsid. And again, we see the decrease in lentiviral population from the starting value, which is a very, very good indication that these capsids started degrading, and then there is a much higher increase in lower molecular weight impurities and the increase in degraded population as well. Increasing it further to 60 degrees, we can see a clear, again, quantify a clear decrease in the population from the starting value even further, and increase in impurities almost 100%. And again, the heat stress damages lentiviral vectors and elevates impurity levels in all of these samples, almost 100%. Now, this is just a summary of all the population analyzed by Caritro. The following to exposure to various thermal stress, we can see a clear trend of our lentiviral vector population decrease, and the very sharp increase in impurity and increase in degraded lentiviral vectors. So besides thermal stress for lentiviral vectors, we tested additional conditions, which is essentially to test how freeze-thaw cycles could potentially impact lentiviral vector population. And in this particular case, lentiviral vectors were pre-diluted in a different buffer and then freeze-thawed. And after that dilution, we measured our lentiviral vectors. And as we can see, so as you can see on this scatter plot, and the gate A, which is in the middle of this scatter plot, there is a decrease of the lentiviral vector population, almost 44% from the starting value. And as we already know from many other researchers, that there is a significant decrease in the functional titer after only one freeze-thaw, but even going even further. And you can see the increase in the lower molecular weight impurities in this case, and that some of these lentiviral vectors were completely degraded, and then they're falling into that gate for low molecular weight impurity. So this has a very important role in the formulation of the lentiviral vectors and finding a stable formulation that will keep lentiviral vectors stable through its shelf life and the life cycle. Now, the instrument that we used in these two experiments are Samux and Caritro, our mass photometer and macro mass photometer, which are our rapid analytical tools for stress testing. And if you can remember from a slide from the beginning of the presentation, that we have a lot of barriers for high throughput scalable testing for quick development. We need something that will allow that quick developability studies, and this is how Samux and Caritro can play that role. First of all, there is a limited sample preparation because these methods are label-free and you can measure them in native state. The results are available in less than 5 minutes, including measurement and analysis, and a very low sample requirement, which means that multiple studies can be performed and not a lot of material will be consumed because only 10 to 20 microliters, around 1E11 particles per mil, are required. It is also important to consider operator costs and scientist costs. It is very easy to use, so it doesn't require a very long training process, because each scientist can be trained in less than a day. Besides the measurement being available in 1 minute, in 1 minute you can see a result. Besides that, the analysis could be fully automated, and that 24 samples take around 1 hour and a half at SAMUX, by using SAMUX auto instrument. Many methods are very expensive to develop, besides consuming very high volumes of material. They do require a very expensive reagents. In this case, that problem is solved by having a solution that is just less than $5 per measurement, and its benchtop instrument does not require lots of space. It is fully optimized for the lab, and it has a small footprint. So Characterise is the second instrument for the characterization of large viruses, including lentiviral vectors and adenoviruses. And it's also single particle analysis, and everything that was listed for SAMUX almost applies for Characterise, which means ease of use of very intuitive workflows for hardware and software. And pretty much once the samples are prepared on the glass slide, and the glass slide put on the instrument, reads it in less than an hour and a half, and samples are immediately ready. So 40 samples that are applied can be then visualized and analyzed quite quickly. So it allows a very high throughput as well as SAMUX. So how to de-risk gene therapy development? So this is where mass spectrometry comes in, because it's a powerful tool to de-risk your gene therapy by providing real-time, label-free insights into product integrity and stability. So it helps you identify vulnerable attributes very early on. So by observing how different stress factors such as temperature, pH, shear, free store cycles can impact the viral vectors, and by testing them using mass spectrometer and macromass spectrometer, we can determine easily which components are most prone to degradation. For instance, as you saw that shift in empty and full population and ssDNA and genetic payload leakage, that gives a straight insight into the failure mechanism that sometimes traditional method may miss. And then how does it help us enhance analytical sensitivity? I'll just take an example of the ddPCR. So sometimes some analytical methods, such as ddPCR, for example, requires incubation time. In this case, for example, by adding a nuclease and having an incubation time at an elevated temperature, maybe could potentially cause the payload leakage. And then by stress testing your samples before exposing them to these conditions that some of these analytical methods require, mass spectrometry can help you optimize other analytical methods. And besides that, it can help you guide a formulation and a process design. For example, mechanical stress is very important for determining which process parameters can be used. And by exposing viral vectors very early on to different types of shear stress, we can determine what type of process parameters can be used, what is a very high flow rate, what pressure should not be utilized during the clarification or the TFF process. So besides that, a formulation is extremely important for the shelf life and the long-term stability products. So mass spectrometry can support like excipient selection by just quickly testing different excipients in the storage under it and seeing whether there's any degradation in the viral vectors happening. So besides the process design and the formulation, it can help informing scientists on storage and handling and logistics, which is also important from the CMC standpoint of view. What is the product tolerance, and are there any temperature excursions that affect the long-term stability, storage conditions, or the shipment of the products that contain viral vectors? And finally, mass photometry delivers data-driven confidence, and that will support regulatory submissions, tech transfer, supporting decisions with mechanistic clarity, and EU with also robust stability data. So in short, mass photometry can help you develop a faster and a smarter choices, and by being very fast and cheap and easy to use, even at very early on in developability stage, but also further down the line in regulatory submission, it could be utilized to deliver safer and more stable gene therapies to patients. And thank you for the attention, and we are happy to take on any questions that you have.
Hena, for your informative presentation. I'm Kevin Flug of LabRoots, and I'm going to be your moderator for today's Q&A. Today's educational web seminar is presented by LabRoots and sponsored by Refeyn. For more information about Refeyn, go to www.refeyn.com. We're now going to begin the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. We'll answer as many questions as we have time for. And I'd like to bring back on Hena Divanovic for Q&A. For a complete biography on Hena, please visit the presenter tab from the menu on the left of your screen. Hena, thank you very much for your time and your presentation. Our first question that we got in is, to what extent can stress testing data obtained via mass photometry be used to inform process robustness studies or in-process control limits during scale-up and manufacturing?
Thank you, Kevin. Thank you for introducing everything, and thank you for your question as well. So to answer to that question, I'll just go back to what it was already spoken about in the presentation. So stress testing with mass photometry can reveal how viral vectors usually respond to those variations in physical and chemical stressors, such as the ones that were listed in shear stress, temperature, pH shift. So, for example, mass photometry can detect and quantify empty fall in partial population. Likewise, macromass photometry, titers, and for lentiviral vectors. Now, the question is how this could be translated into process by identifying which parameters affect the vector stability and integrity the most, and what is going to be translated to the scale-up. So, for instance, during the scale-up, changing the process parameters or exposing it to different shear stress could help researchers discover which conditions negatively impact them, what is degrading the capsid, and then based on that define the right ranges and establish process control limits very, very early on. And the good thing with mass photometry, that is possible to obtain in just minutes.
Awesome. Thank you very much. The next question we received asks, have you observed differences in stress susceptibility between various AAV serotypes?
That's a great question. So in the presentation, we only show one serotype. And so AAV serotypes are inherently different, and they usually exhibit different characteristics. So some researchers even say that thermal characteristic and stability are essentially the fingerprint of AAVs. So although it's not shown in this presentation, when different serotypes are exposed to the same temperature as the one that was shown in the presentation, AAV8, they don't behave similarly. So some of them are degrading at a faster rate at the lower temperature, some of them at the higher one. But the good thing that mass photometry is a label-free method, and in that case could detect all of those shifts. So I did test different serotypes, but it's not presented here. And stay tuned, more data will be coming from Refeyn on this.
Awesome. Thank you very much. Looks like we've got time for one more question. And the question that came in was, do you think micromass photometry could be used to monitor lentiviral vector stability during formulation?
Yeah. Thank you for the question. So, yeah, I think macromass photometry could be very valuable. I know that we just touched on that very briefly with one example that was listed there, where lentiviral vectors were diluted into a different formulation buffer. So why is it useful? Because it's fast and relatively inexpensive, and you can see those changes by changing or exposing viral vectors to different formulation. So when the researcher needs to screen many buffer and excipient conditions to see which one is the best to preserve vector stability and integrity over time or over shelf life, it's useful to use macromass photometry, because these studies then can be done on a very, very small scale, and it's ideal to compare multiple formulation side by side. And that is a very early indication which condition stabilizes the vector before moving on to more like detailed and resource intensive formulation studies.
Awesome. Well, thank you again, Hena, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, Refeyn, for sponsoring today's webinar. Please visit Refeyn's booth in the exhibit hall after the webinar for more information and resources. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year, so that'll give you until October 15th of 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So, Hena, once again, thank you for your time. Thank you for your audience. And until next time, goodbye.